Hi everyone, today we'll be covering the IGCSE Human Biology past paper. This is from May-June 2023, and this is paper 1. You are given 1 hour and 45 minutes to complete this paper. This paper is worth 90 marks, as you can see here, and this paper tests on the entire syllabus. All right, let's get started with the first question. All right, use information from the box to complete the sentences about the movement of molecules. All right, so molecules can move in and out of cells by osmosis, diffusion, and active transport. So the first part, it says osmosis is the movement of, and this is water, across a partially permeable membrane, or in this case, they have the word selectively permeable membrane, so we'll just use that. So I'll just include a diagram over here to showcase what osmosis is. So you can see on the left you have a high concentration of water, or you can think of it as a dilute solution, and on the right we have a more concentrated solution. And you can see this gray dashed line here represents the partially permeable membrane or the selectively permeable membrane. And you can see water is moving from a region that's more dilute to a region that's more concentrated. All right, let's remove this. And let's talk about diffusion. So diffusion allows the passage of molecules. And then the next part says something, a concentration gradient. So it's going to be either against or down a concentration gradient. In this case, diffusion is going down the concentration gradient. So what that means is you're going from a region of high concentration to a region of low concentration. You could kind of think about it as rolling something down a hill. The speed of movement is affected by the surface area to volume ratio of the cell. So that's why cells like your intestinal cells, right? They've got a lot of microvilli, and that's to increase surface area for absorption so that your intestines can absorb nutrient molecules that are in your intestine. All right, and the third part is active transport requires the use of energy stored in the form of ATP. So ATP is our sort of energy currency and allows for molecules to move against. You can see that there. Against a concentration gradient. Let me show you a diagram of that. All right, so you can see an example here. So we have low concentration on the outside, high concentration on the inside, and it goes from low to high concentration using energy. So it's like rolling something up a hill. All right, let's move on to the next question. The photograph shows alveoli in the lungs. Alveoli are the site of exchange for gases between the air and the blood. Part A1, state in the correct order the structures that air must pass through from the atmosphere to the alveoli. All right, so I've included a diagram here. In order for the air to go to the alveoli, first it must go through the nasal cavity, so we're breathing that in. And then it goes to the trachea next. So that's basically our windpipe. And then there are two of these branches because we've got two lungs. We refer to that as our bronchus. And then the bronchus further branches out to form bronchioles. And at the end of the bronchioles, we have the alveoli. All right, so let's just write that down. We have the nasal cavity, or you can also say nose or mouth, that's fine too. Then the trachea, the bronchi, then the bronchioles. The reason why I put bronchi here is because that is plural. All right, part 2, state 2 features shown in the diagram that allow the efficient exchange of gases. All right, so here we have the alveoli. All right, so for all gas exchange surfaces, it's very important that you have very thin walls, and you can see that here. Or you can say very thin epithelium. The reason why is you want to reduce the distance traveled for these gases, right? So you don't want the gases to travel that far. Number 2, the alveoli has a very large surface area. So instead of having an alveoli that looks kind of spherical, you instead have an alveoli that sort of protrudes out like this, as you can see the diagram that I've just drawn. So that is important in order to increase the rate of diffusion of these gases. All right, let's write that down. So number 1, it has thin epithelium, or you can say thin walls. And number 2, it has a large surface area. Okay, let's move on to part B1. Which is the percentage of carbon dioxide in the atmosphere? And that answer, you do have to remember, it is 0.04%. Okay, part 2. The table shows the results obtained when a person at rest breathes in air that contains different percentages of CO2. Calculate the volume of air in dm³ breathed per minute. Let's write that down. When the person breathes in air containing 3% of carbon dioxide. All right, so let's look back at the table. So the term tidal volume refers to the volume of air that is breathed in and breathed out during a normal breath, right? And we know that because we're looking at 3% of CO2, we're looking at this column. So imagine for each breath, this person is inhaling and exhaling 1,200 cm³. And they're doing this 16 times per minute. So we would do 12,000 times 16. And that should get us 19,200. But remember here, they want us to calculate this or convert this to dm³. Right now we have cm³. And in the square brackets over here, they give us the conversion rate. So we know that 1,000 cm³ equals 1 dm³. So in order to convert it to dm³, we would divide that by 1,000. So that would be 19.2 dm³. All right, 19.2. All right, this part, the air a person breathes in contains 20% of oxygen. The body uses 20% of this oxygen. Calculate the volume of oxygen used per minute when the person breathes in air containing 1.6% of carbon dioxide. Okay, so let's scroll up first. I'm going to remove the annotations on this table so it's not too confusing. All right, so first we want to calculate the total volume of air breathed in for that person at 1.6% of carbon dioxide. So now we're looking at this column. So let's write volume of air breathed in. And again, for one breath, the tidal volume is 700 cm³, and then they're breathing in 15 breaths per minute. So we would do 700 times by 15, and that should give us 10,500. Let me write cm³. Okay, and then it says the air a person breathes contain 20% of oxygen. So let's do volume of oxygen. So that would be 20% or 0.2. times by 10,500. And what would that be? 0.2 times 10,500. That would be 2,100 cm cubed. And lastly, the second sentence is the body only uses 20% of that oxygen. So volume used by body. So we would do 20%, so that's 0.2 times 2100, and that'll give us 420 cm cubed. All right, 420. Let's scroll to the fourth part. Explain the effect of increasing the percentage of CO2 in the air a person breathes in on the tidal volume and rate of breathing. So I've included a diagram here, but let's go back up to the table. So we can see that as the percentage of carbon dioxide increases, so does the tidal volume and the rate of breathing. So let's go back down to this graph. So this flow chart shows how breathing rate is controlled in our body. So it's very important that if we have a lot of carbon dioxide in our blood, our body needs to remove that carbon dioxide. And in order to do that, we need to increase our breathing rate and increase the tidal volume. So in order for our body to do that, first, the carbon dioxide level needs to be detected by our body. And it does this using receptors. And you can see that they're specifically chemoreceptors. And you can find these chemoreceptors in the medulla, so that's in the brain, and the arteries. So specifically in your syllabus, it says the aorta and the carotid arteries. So when these chemoreceptors detect high levels of carbon dioxide, it sends a message to the respiratory center in the brain. And your respiratory center sends a message to your diaphragm and your intercostal muscles in order to increase the breathing rate. So you're able to remove the carbon dioxide via your lungs. Okay, so this question asks us to explain it, so we can say when there are high levels of carbon dioxide in the blood, gets detected by chemoreceptors in the medulla and arteries. And then we can say increases the breathing rate so that more carbon dioxide is removed. All right, let's go through the marks. The first mark is when there's more carbon dioxide in your blood or in your body. The second mark is detected by chemoreceptors in the arteries. The third mark is, you can say, an increase in the breathing rate, or you can say the tidal volume increases. And lastly, more carbon dioxide is removed. All right, let's move on to question 3. A student investigates a reaction. The reaction is catalyzed by an enzyme and produces a gas. The student collects the gas for 10 minutes and measures the volume of the gas. The student repeats this every 10 minutes for 1 hour. All right. Okay, so part A1, it says add a suitable labeled scale to the x-axis. So we can see in the first paragraph that it says that the student collects the gas for 10 minutes. That means from 0 to 10 minutes, they'll collect the gas. Then 11 to 20 minutes, they'll collect the gas, and so on, right? And the student repeats this every 10 minutes for 1 hour. And we can see there should be 6, right? 2, 3, 4, 5, 6 sort of time periods in which they are collecting gas. So let's remove that there and let's do time in minutes. And then we can do 0 to 10 minutes, 11 to 20 minutes, 21 to 30 minutes, 31 to 40 minutes, 41 to 50, and lastly, 51 to 60 minutes. All right, estimate the total gas produced after 30 minutes. Okay, so in this part, we're trying to add the total volume for these 3 bars, right? So the first one, that looks like it's about 75. The second one looks like it's about 72. And the third one, it's below 70, so maybe it's about like 67 or 68. And they do give you a bit of a range here. So in the marking scheme, the first bar that you see there, the one that says 75, that has a range of plus or minus 2. So for example, the first bar has a range of 73 to 77. And this range, this plus or minus 2 range, applies to all 3 of the bars. So let's add everything up together. 75 plus 72 plus 67, and that should give us 214. So remember, the marking scheme does give you a bit of leeway there. Okay, let's move on to part 3. Describe the trend shown by the results. All right, so the question says describe, which means we're supposed to talk about what we see in the graph. So let's scroll back up. I'm just going to erase this so we can see this properly. So the volume of the gas produced from this reaction is indicative of the rate of reaction of this enzyme. So the more gas that is produced in that time frame, the greater the rate of reaction. So when we look at the graph, we can see that at the beginning, the largest amount of gas is produced. Then for the next 10 minutes and onwards, there is less gas that is produced. We can even comment on the fact that after this point here, right, they're producing less than half of the volume of the original amount of gas. So after 40 minutes, there is little gas that is produced. All right, so let's go back here. We can say the beginning of the reaction, the largest volume of gas was produced. For the next gas production decreases. And then we can see after 40 minutes, a lot less gas is produced. All right, let's go through the marks. The first mark is large amount of gas produced in the beginning. The second mark is the rate slows, or there's less gas for the next 10 minutes. And then the third mark is little gas is produced after 40 minutes. All right, let's go to part 4. Explain the students' results obtained in the investigation. So in this part, in the beginning, you would have a lot of substrate, and therefore there would be more collisions with each other and more enzyme substrate complexes forming with each other. And as the reaction progresses, less and less substrate is available. And because there's less substrate available, there is therefore less gas produced. So we can say at the beginning, there was a high concentration of substrate. Okay. Probably not, however. As the reaction progressed, less substrate was available. And therefore, the rate of reaction decreased. Reaction decreased. All right, so let's go through the marks. The first mark is there was a high concentration of substrate in the beginning, or you can say many enzyme substrate complexes were formed in the beginning. And then you can say as the reaction progresses, there's less substrate available, and therefore it resulted in the rate of reaction decreasing. All right, let's move on to part B. Explain the effect of the results if the student had used a competitive inhibitor at the start of the investigation. So a competitive inhibitor is a kind of inhibitor that competes for the active site, and you can see that there. So it competes for the active site and blocks the substrate from forming the enzyme substrate complex, and therefore no reaction occurs. So this question is worth 2 marks, and we're supposed to explain the effect of the results. So we can say, well, if you have a competitive inhibitor, then you'd have less gas produced because the inhibitor would be blocking the active site and prevents the substrate from binding to it. If there was an inhibitor, less gas would be produced as the inhibitor binds to the active site and blocks the substrate from binding to it. All right, let's go over the marks. So the first mark is less gas would be produced, and the second mark would be the active site is being blocked. All right, let's go on to question 4. All right, it says the eye contains receptors that can detect light. Name 2 other senses. So this is referring to your 5 senses, right? Seeing, touching, hearing, tasting, and smelling. So we can name any of the 2. So let's write tasting and smelling. All right, the next part. The table shows how the focal length of the eye lens changes as the thickness of the lens changes. Part 1, describe the relationship between the thickness of the lens and its focal length. All right, so we can see according to the table, as the thickness of the lens decreases, the focal length increases. Another thing you can also say is as the thickness of the lens decreases, the interval between the focal lengths increases. So we can see from 12.5 to 14, that's a difference of 1.5. From 14 to 15.5, that's a difference of 1.5 again. But from this one, 15.5 to 18, that's a difference of 2.5. This is a difference of 3. This is a difference of 4. So the intervals get larger as the thickness of lens decreases. Another thing that's mentioned in the marking scheme is that these 2 variables are inversely proportional. So it says in the marking scheme, when these 2 variables are multiplied, it gives you a constant. And we can see that here. So for example, if you times 10 and 12.5, that gives you 125. If you times 9 and 14 together, that gives you 126, which is quite similar. If you times 8 and 15.5 together, that gives you 124. If you times 7 and 18, that'll give you 126. So you can see the numbers are quite similar. I'll just write down the first 2 points that I mentioned earlier. So as the thickness of the lens decreases, the focal length increases as the thickness increases, the intervals at which the focal length increases gets larger. All right, let's do part 2. Give the effect of the diameter of the lens as the thickness increases. So we'll go over a couple of diagrams first, and the next question I'll go over it in more detail. So here we have the eyes, and when we're looking at distant or near objects, the shape of our lens changes. So at distant objects, the lens gets pulled by the suspensory ligaments, whereas in nearby objects, the lens is able to thicken. Now, which brings us to, let me just remove this. So we can see when the lens is flattened, we can see that the diameter is larger. But when the lens becomes thicker, right, the diameter decreases. All right. So we can write as the lens thickens, the diameter decreases. Okay, let's move on to the third part. A person is looking at a distant object, then looks at a near object. Use the data in the table to explain the changes that occur in the eye so that the person is able to form an image of the near object on the retina. All right, so this question is talking about a near object. So rays that are coming off of a near object would have diverging rays, as you can see there, whereas if it's a distant object, the lines are parallel. So if the rays are diverging, that means that you would need to bend the light more. So the lens needs to be thick. So for the lens to be thick, two things happen, and we can see that here. Number one, the ciliary muscles, they need to contract. And what happens is that causes the suspensory ligaments that you see here to become slackened or to relax. So the thick lens is now able to refract the light a lot more. And we can see that as the lens gets thicker, right? So here we have 10, the focal length decreases. And the focal length refers to this part here, the length between this and this. So we can say light rays that are coming from a near object are diverging. Therefore, the lens needs to be able to refract the light more. To do this, the ciliary muscles contract, which causes the suspensory ligaments to slacken. This causes the lens to become thicker. So that the focal length decreases. All right, let's go over the marks. The first mark is rays from a near object are diverging. The second mark is ciliary muscles contract. Third mark is suspensory ligaments slacken. 1, 2, 3, fourth mark is lens becomes thicker. A fifth mark you can say is so that it causes the light to refract more. And then you can also mention how the focal length decreases. All right, let's move on to the next question. Part C, cone cells are found in the retina of the eye. They detect color. The graph shows the absorption of different wavelengths of light by 3 types of cone cell. All right, the first part says determine the wavelength at which a red detecting cone absorbs the most light. So the red detecting cone would be the dotted one that we see here, and this would be where the peak is, and we can see this part is 550, 560, 570, so that's about 575. Let's write that down. 575. Determine the maximum difference in the wavelengths of the light that can be absorbed by the blue detecting cones. Okay, so the blue detecting cones are the solid line. So we can see it goes from, it's this line that you see there. You can see it goes from 400 to 1, 2, 3, so that's 530. So 530 minus 400 gives you 130. So let's write 130 nanometers. Suggest why it is better to have 3 types of cone rather than just 1 type of cone. So we can see from the graph, each cone absorbs different wavelengths, and if you can absorb different wavelengths, then you would be able to see different colors. So the 3 cones absorb different wavelengths. Therefore, more colors can be detected. That's the end of the video. If you want to look at question 5 and onwards, you can click for part 2 in the description below. All right, bye.